Um, Great. So thank you all for coming. I am excited to introduce Jess Loeffler to you. She is a graduate student at CU Denver and is with us here at Denver Botanic Gardens. She came to me initially through Zoom over the COVID years because she was currently living in Indiana and her partner had moved to Denver. And the idea was that they'd both relocate to Denver, and her thoughts were I could go back to grad school since I just finished my undergrad in Minnesota, and she was working on plant pathology related stuff. And she'll give you some more of a background, but she approached me because she wanted to go to grad school, basically because she was still interested in fungi. And so I said yes. And of course, we kind of waited around till the opportunity presented itself. And then she joined my lab, and we were bouncing around between different ideas as to what she could study. And a colleague of mine, Amy Honan, actually came to me with this idea for this project. And it's a pretty cool and interesting project because it deals with plant-fungal interactions and kind of explains and gives us an understanding of fungi are important to our environment and to our ecosystems, especially in terms of their relationship to plants, which kind of explains why we here at Denver Botanic Gardens are interested in fungi, not just plants. So with that, I'd like to introduce Jess Loeffler, and she will uh, explain her project to you and what the whole biological situation is. So come up, Jess.
Let's see if I can figure out how to do this. Does that look? Did I do it? Awesome. Okay. Hi. Like Andy said, I'm Jess, and my talk is mainly focused on mycoheterotrophic plants and the fungi that feed them. So Andy gave me a really good introduction. I will also briefly introduce myself if I can move my slide. Great. Hi, I'm Jess. Like Andy said, I'm a master's student at CU Denver with the Denver Botanic Gardens. My previous research experience has mostly been with plant pathology, so we're fungi, we're the bad guys. And that was another reason this project was really cool, is because it was finally a project where fungi weren't the villains, right? So like Andy said, I started getting in contact with him when I lived in Indiana. When I moved out here, I didn't go to grad school right away. I realized that I had a gap in my resume. I'd never done any molecular work before. And so I worked at a virology lab. That was a weird little side tangent. That's actually what the video up at the top right is those are human nasal cells. Maybe I should go. Those are human nasal cells. And if you see them moving, they have little like cilia at the top that just kind of like beat mucus back and forth. Anyway, I just thought that was a fun little video. But yeah, so once I moved here to Denver, I was working in this human virology lab, but I wanted to still stay in the plant fungal realm. And so I started coming to CMS meetings, and that's where Andy started talking to me more seriously about like volunteering with Denver Botanic Gardens. And now I'm a grad student. During this talk, I'm going to go briefly over kind of the way nutrients move through the ecosystem, right? So we're going to talk about mycorrhizal fungi and then get an overview of what mycoheterotrophic plants do, what they are, and then which fungi feed them. And that'll bring me into what my research question is, my hypotheses, how I collected my data, and then what results we have so far. I'm not quite finished with the analysis yet, but I do have some pretty cool results. So the first question I have that feels a little bit weird to ask at a mycological meeting first is, how do plants get food? And the answer is photosynthesis, right? Plants take carbon dioxide, they take water and light energy and make sugar and oxygen. But if you've tried to go grow plants before, you know that they don't just need light and air and water, right? Like they also need nutrients. Those nutrients, the big ones are like nitrogen, phosphorus, potassium, but they also need other micronutrients. And they're going to get those nutrients via their root system. So they're going to uptake those along with the water. A really important aspect of how they uptake these nutrients is actually mycorrhizal fungi, right? So this is where the fungi are big and important. Fungi will provide water and mineral nutrients, and in return get sugars from the plant. And so if we look at this picture, we can actually see that the plant roots end like right about here. All the rest of this is mycorrhizal fungi. And this is a hugely important symbiosis, both for plants and for fungi, right? For the plants, 80% of plants on this planet participate in this symbiosis, and thousands of fungi participate. This is a huge way, like forests, the way we know them, wouldn't exist without these mycorrhizal fungi. All right, so we figured it out. We figured out plants get their energy from photosynthesis and mycorrhizal symbiosis, but not all plants. Some plants can be parasitic, and some plants can parasitize other plants. If you've been out hiking, you might have seen dwarf mistletoe. This is a plant that will parasitize lodgepole pine and get all of its nutrients from the pine. Like, it doesn't photosynthesize at all. It's just getting all of its energy from the tree. So plants can parasitize plants, but they can also parasitize fungi. And the term that we use for that is mycoheterotrophy. So really broadly speaking, mycoheterotrophy is just like a really big fancy word for uses fungi for nutrition. Actually, I'm pretty positive that there's a lot of people out in this audience right now that are mycoheterotrophs, right? But in this case, we're going to be talking about mycoheterotrophy in the plant community. Within plants, mycoheterotrophy is actually relatively common. It's evolved at least 17 different times, or 50 times in 17 plant families. The examples that I have up here are just a small sample of the diversity that we see. There's this Pterospora andromeda, the pine drop, we can find here in Colorado. There's this orchid, this Epipogium roseum. I believe that one's found in Europe. And then this Voirea tenella, another mycoheterotroph that you can find like in Mexico, right? They all look very different from each other. They're very diverse. Mycoheterotrophy is in general hypothesized to be an adaptation to light scarcity, right? So if I'm a plant, I'm pretty little compared to trees, right? And it's going to be really hard for me to compete with that light energy that we decided was so important earlier, right? And so rather than compete with trees for the light and the energy above them, they've decided to tap into the energy below. And where are the fungi getting this energy from? In general, there's kind of like two camps that mycoheterotrophic plants tend to parasitize. We have mycorrhizal fungi, which we talked about earlier. That's that symbiosis. They can also parasitize saprotrophic fungi. So these are fungi that are going to be breaking down dead organic material. So mycoheterotrophs can get energy from both of them. Now, I think the main thing that I want to stress with this is that the thing that makes them mycoheterotrophic is the fact that they're getting energy from the fungus. This tree wouldn't count as mycoheterotrophic because it's not taking energy, it's providing it. And the mycoheterotrophic plant is kind of like an epiparasite off the tree in a sense, right? Because it's taking the energy from the fungus, but that energy came from the tree. Parasitism with extra steps, right? Okay, wanted to take a quick second just to see how we feel with mycoheterotrophy versus mycorrhizal symbiosis. I think we'll just do like thumbs up if we feel good. Sweet, love it. Okay, because I'm going to make it more complicated now, because not all plants parasitize fungi to the same degree, right? This is a figure I've been using to indicate the kind of like wibbly wobbliness of mycoheterotrophy. So all the way on the left here, we've got full autotrophy. These are plants that are not utilizing energy from fungi. All the way on the right, we've got full mycoheterotrophy. These are plants that are only getting their energy from fungi. And in the middle, we've got this group called the partial mycoheterotrophs, which they can still photosynthesize, but they're still taking energy from fungi. There's kind of a fourth category that doesn't fit well onto this spectrum. It's called initial mycoheterotrophy. This is where fungi are actually required for a seed to germinate. A group that does this pretty commonly would be orchids. So they make these like itty teeny tiny baby seeds that require energy from fungi for the seed to actually have enough energy to sprout, to germinate, to get started. Yeah. And the reason it doesn't fit well into this spectrum is because plants that are initially mycoheterotrophic can grow up to be autotrophic. They can grow up to be partially mycoheterotrophic. They can also grow up to be fully mycoheterotrophic. So I haven't included them on this end. For the rest of this talk, we're mainly going to be focusing on what the plants are going to be doing once they are at maturity, once they've grown up. So getting into each stage specifically, full mycoheterotrophy is the use of only fungi for nutrition. And just looking at them, they look weird, right? Like they're not green. If you're just looking at this plant, you're just like, this is either like a really sick plant or something funky is going on here. Before we had the tools to understand that they're actually getting their energy from fungi, these plants were believed to be saprotrophic themselves, right? People thought that they were decomposer plants, but in reality, they're getting their energy from fungi. So they don't have chlorophyll because they don't need to photosynthesize. They don't have leaves. Again, no need for photosynthesis. And their root systems look pretty funky because this is where those interactions are happening. This is where the fungus is interacting with the plant roots to give the plant energy. And these examples here are all, this is Pterospora andromeda. This is a pine drop. We can find this in Colorado. It's actually pretty common out here. This one in the middle is Sarcodes sanguinea. This is snow plant. This is a plant that I found out in California. It's pretty common out in the Sierra Nevadas. And this last one is Monotropa uniflora, or the ghost pipe. This is one that I found in Canada. A lot of times they'll grow in little bunches. Yeah, they're very cute and they're very pretty. So which fungi are they getting their energy from? These full mycoheterotrophs are actually picky. So despite the fact that they're getting all their energy from fungi, they're specific about which fungi. These two plants in particular are incredibly picky. The pine drop only associates with we only know it to associate with 3 species of Rhizopogon, which is crazy, right? There's a ton of genera of fungi and a bunch of species within each genera, and this one is only associating with 3 that we know of. That's crazy. And the same thing with this snow plant. It's only known to associate with 2 species of Rhizopogon. And now moving into partial mycoheterotrophy. So these are plants that kind of have a foot in both camps, right? They can do both. They can photosynthesize, but they're also supplementing their energy requirements through the use of fungi. These ones look like normal plants, right? You kind of wouldn't think that anything's up with these guys. They have regular old green leaves. They have regular old root systems. These are 2 examples in the Ericaceae family. This is Pyrola acerifolia, bog wintergreen, and this other one is Ortholia secunda, or the side bells wintergreen. You can find these in a lot of places in the mountains. And they're pretty unsuspecting, right? They're small. They don't look super special, but they are special. And as for which fungi they associate with, they're at the very least less picky. So this was a study done in Japan looking at 2 species of Pyrola. This circle are the fungi that were found on root tips from oak trees. So I put little symbols to show us like where they fall on the spectrum of mycoheterotrophy. So this one would fall here into full autotrophy. The other species that they looked at was Pyrola japonica. This is the partial mycoheterotroph. So that goes here, and it's associating with fewer fungi. And the last plant that they examined was Pyrola subaphylla. This is a pyrola that is really close to being fully mycoheterotrophic. Its leaves are teeny tiny. They're kind of not pulling their weight. This one is very close to being fully mycoheterotrophic, and that associates with the fewest fungi. And so that led us to the question of, does an increase in mycoheterotrophy lead to increased fungal specificity? So our hypothesis looks like this, right? Where we've got the spectrum of mycoheterotrophy and then this idea of like general plants that'll associate with kind of any old fungus. And then as it goes along this gradient, it starts getting more and more specific the more it relies on fungi. So that's my whole research question is, can we see this in mycoheterotrophic plants? So how do we test this? Well, I'm a master's student and I don't really feel like being here for 20 years. So we need to pick a model, right? The model family that we chose was the Ericaceae family of plants. And this is a really cool family all on its own, right? The core Ericaceae, we've got like blueberry rhododendrons, and those are mycorrhizal, right? They actually have their own special group of mycorrhizal fungi called the ericoid mycorrhizal fungi because they're very special. But in this family, there's also the monotropoid family. So a bunch of those pictures I showed you before, like the pine drops, the snow plant, the ghost pipe, those are all in this group right here. And these are the ones that had those very specific relationships. In the middle, we've got this little family called the Pyrroloideae subfamily. In this family, plants can exhibit an array of mycoheterotrophy. Some of the plants at maturity are autotrophs. Some of them are partial mycoheterotrophs. There's even one full mycoheterotrophic plant. But all of them are initially mycoheterotrophic, which if you remember, that means they just require fungi to germinate. So within this group, I chose two species. I chose Chimaphila umbellata and Pyrola acerifolia. So they're both in that same subfamily. They both have green leaves. They're both initially mycoheterotrophic, so they require a partner to germinate. And they share similar habitats and ranges. I could find them in the same places. And that was also going to help us figure out, like, compare the fungi to each other. On the spectrum, Chimaphila umbellata falls into it's a fully autotrophic plant. The reason I put the dot so far to the right is because it is initially mycoheterotrophic, and we consider that starting to be close to partially mycoheterotrophic. But then Pyrola acerifolia is partially mycoheterotrophic. And we wanted to see, using our hypothesis from earlier, do we see that Chimaphila associates with more fungi compared to Pyrola? So to go out and sample, I went to 3 states. I did some sampling. Oh, I should explain this. So the black dots in this are iNaturalist observations. On the top, we have iNaturalist observations of Pyrola. On the bottom, we have iNaturalist observations of Chimaphila. The red dots are where I went and sampled plants. So we sampled in Minnesota, Colorado, and California. In Minnesota, I was originally going to sample from one site, but I had the darndest time finding Chimaphila. There were 2 days in the rain with no plants. It was a bummer. So I kind of have 3 sites in Minnesota. I've got Itasca and Lester Lake, which is where I found Pyrola, but no Chimaphila. And then Boulder Lake, where I started to find some Chimaphila. In Colorado, I went to Meyer Ranch State Park, Fraser Experimental Forest, and the Mountain Research Station. And in California, I went to the Sierra Nevada Field campus. And you'll also notice that for the Minnesota and California populations, I only collected plants before and during flowering. I wanted to sample multiple times per year just because those mycorrhizal relationships can change over the course of the growing season, and we wanted to get the best idea of which fungi they were associating with. In Colorado, I did before, during, and after flowering. That was mainly convenience, but also previous literature indicated that the later into the growing season that you go, the fewer fungi these plants are associating with anyways. So my sampling scheme, at each site, I set up 2 plots, 2 populations of each plant species. So I would grab Pyrola and Chimaphila, and I would also grab soil from nearby Pyrola and Chimaphila plants. We wanted to grab soil so that we had a better idea of what fungi were available to the plants, just so that we could say, like, in the end, okay, Chemophila had the same fungi available to it, but it was associating with more, and that would be more indicative of what's going on, other than just like, oh, hey, this is what we found in the plants. No idea what was available to them. This top one is what a pyrola looks like when we pulled it up. And this middle one is what Chemophila looks like. And so in each plot, I actually grabbed 3 plants. So the problem with these guys, as you can see, they've got these this rhizomatous root system, which means they kind of spread all over and send up green shoots, which made it really difficult to tell what was an individual plant. So when it came down to the sequencing, I actually just pooled the 3 plants together into one, just because I wasn't able to tell what an individual was anyway. The only way I would have been able to tell is if we like ripped up the whole forest floor and like followed the roots around. And I really didn't think that the people who issued me my permits were going to be cool with that. So yeah, once we collected the plants, I would sample and pluck off these little itty bitty baby rootlets right here, because that's where those mycorrhizal connections were going to happen. Put them into an Eppendorf tube, dried them out, I extracted the fungal DNA, and I did the same thing with the soil, where I extracted fungal DNA. The next thing we did, I used a metabarcoding approach. So that basically means like I was taking one specific region on the genes to compare to other sequences. I used the ITS2 region, mostly because it's like one of the more common ones to use, and we had done it before in the lab. I used different databases to blast my sequences to. That basically just means compare my sequences to reference sequences. So we can be like, okay, those are very similar. That's probably the same thing. That would assign an identity to each sequence that I got. So once I got those identities back, I was looking at a spreadsheet with 1,472 different fungi, trying to figure out how the heck I was going to analyze this data, because that was pretty overwhelming. And there was this great database built by Nguyen et al. In 2016 called Fungild that allows me to compare. So it's like I could put in the name of a fungus and it would tell me what it does. Does it decay wood? Is it a pathogen? Mycorrhizal. So once I finally had that together, I could start my analysis. Now, interestingly, a lot of my sequences didn't match any of the references perfectly. So just over half did. I got sequences that were 90, I used the threshold of 97%. So if my sequence was 97% similar to my reference sequence, I was able to say, yep, that's it, looks good. It'd be like if you had a barcode and you scanned it and it told you that is absolutely Fruit Loops, you know? But for the ones that weren't 97% similar, I basically told it, you know, we could scan the barcode and it would tell me this is cereal, but I can't tell if it's Fruit Loops or Cheerios. So this is just kind of a plug for Fungaria. It's important to document and sequence collections. Okay, so getting into my data, I wanted to first start us off with what I expected to see. So this is, I'm going to present data using an NMDS, which is non-metric multidimensional scaling, which that name doesn't really help. Basically, what it means that in taking complicated data and squishing it onto an easily readable graph. If points are close together, that means they are similar. If points are far apart, they weren't that similar. Each point on this graph is going to indicate like one Eppendorf tube of sampling. So that's going to be one collection of 3 plants from each plot within each site, within each species. So if my hypothesis was supported, I was expecting to see something like this, right? Where the Chemophila umbellata were going to be pretty different from each other, and the pyrola fungal communities were going to be similar. If my hypothesis is not supported, I anticipated seeing something like this, kind of a jumbled mess. We can't really pull any patterns out. Nothing. What I got was neither of those. So in fact, the fungal communities on Haemophila were far more similar to each other than communities of Pyrola. So everything in a circle here, or everything symbolized by a circle, is a collection of Haemophila umbellata. Everything in the triangles is that Pyrola acerifolia. And I'm going to show you one more figure, and it's the same thing. I just colored it differently and put different circles around it. So this is the same figure, but this circle is indicating plants that were collected in Colorado, specifically Pyrola that was collected in Colorado. This kind of more purplish color was Pyrola collected in California, and this yellow was Pyrola collected in Minnesota. And it's the same colors for Haemophila. Each color of the dot is just one of the sites I visited. It's not really important where those sites were or what they were. I mainly wanted to color it to highlight some of the clustering by Haemophila. So with Haemophila, we've got, these were all collected from one site, this kind of like teal green, and they're really close together, right? Those fungal communities were really similar to each other. But in Pyrola, if we look at the same Pyrola collected from the same plot, it's right here, we've got some green over here. There's some of that same green over there. They're really different from each other. I mean, heck, these Pyrola collected in Minnesota, they're not even similar to any of the other Pyrola I collected. I also included this like statistic, which was basically trying to confirm that I wasn't crazy using numbers. It was a test that looks at the latitude and longitude from each collection and compares, like basically looks at this NMDS plot as just like, yes, with Haemophila umbellata, we see more similar communities when in more closely, geography does impact what fungal communities that we see. It impacts them for both, but the effect is stronger in Haemophila. So that kind of busted my hypothesis, right? Kind of takes it from looking like a really nice triangle into whatever that is. But let's keep looking because I haven't even looked at what fungi I got back yet, right? When I used that data I got back from Fungild, we're able to see that proportionally, Pyrola acerifolia is actually associating with a higher proportion of ectomycorrhizal fungi compared to Haemophila. So with Haemophila, there's like pretty even dispersal between ectomycorrhizal, some saprotrophic fungi, and then some plant pathogens. But with Pyrola, there's this, it's nearly 50% of the fungi that we found on the roots were ectomycorrhizal. And if we zoom into that even a little bit further, we can start to pull out like what genera of fungi we were finding on each of these. And a lot of them aren't the very recognizable ones, right? So when we're just looking at similarities, there was kind of a similar proportion of Hyalocypha, this Odiodendron, Russula, and Tomentella. This is what Hyalocypha looks like. It's just these really adorable little cups. So that was found in kind of similar proportions on both. In Haemophila, there was a larger proportion of Odiodendron, but Pyrola also had a large amount of it. And that kind of looks like this. Both associated with similar amounts of Russula. Hopefully we're a little bit familiar with Russula. And then lastly, Pyrola associated with a bunch of Tomentella, especially in comparison to Haemophila, but they both associated with a bunch. And it's this kind of, Tomentella are really cool. They're really diverse. They just haven't been really thoroughly studied because they look like that. They're not the fun ones to look at. But clearly they're very important. All right, now on to the differences. So which ones were different? The ones that I picked out that were in one but not the other, we saw some Cadophora in Pyrola. That looks like this. Again, not super fun. There was this Hymenocyphus that we found in Pyrola, but not Haemophila, and that looks like this. We also found this Pesloma in Pyrola, but not in Haemophila. if you're looking at that, you're like, you messed up. You put the same picture on there twice. I unfortunately did not. So when I was Googling these pictures, it came back that there's Pezloma. Pezloma and Hemenocyphus are likely the same organism. One of my databases just had a different name from the other. So that's a fun problem that I'm gonna figure out how to solve, because this could potentially be inflating the diversity of pyrola, right? Like if we've got different names on the same fungus, that might make it look, that might increase the amount of diversity that we see artificially. And lastly, on Chemophila, but not pyrola in a turn of events, we found Hyphidiscus, which is this, again, not super exciting, but kind of little crusty cup. Okie doke. So what has that told us? Well, my hypothesis was wrong. But there is, in general, a lot of diversity associated with both plants. It looks like there is likely a higher proportion of mycorrhizal fungi associated with Pyrola acerifolia, but Chemophila umbellata may actually be more specialized compared to Pyrola, which is exactly the opposite of what I anticipated. So what the heck is going on here is the question I've been asking myself for, well, since I started looking at this data. And some of the possibilities I thought of were, since Pyrola requires more input from fungi, it might be kind of recruiting more broadly, trying to satisfy its nutritional needs. Another possibility is that Chemophila itself actually has more specific microhabitat requirements, right? So I mentioned that I had a pretty tough time finding Chemophila compared to Pyrola. So there's a chance that it has different habitat requirements that aren't fungal related, but are reducing the amount of fungi found on those Chemophila. So how do I figure this out? One of the things I plan on doing next is to not only just examine like the list of fungi, but look at how those fungi are related to each other. There is a method that you can use where you create little phylogenetic trees of your samples and look at how those fungi are related to each other and compare them to another sample. And so it would be very similar to those plots I showed you at the beginning, where points that are close together have fungi that are closely related to each other, and plots that are further away would have fungi that are related to each other. The other thing that I really, really want to do is compare the results that we see from the roots to what we find in the soil, right? So I mentioned that Chemophila might be having some of those like microhabitat differences. This would help us figure out, this would see like, okay, what were the fungi that were available to these plants versus what are they actually associating with? And so for everyone to help me with this research, I want to give some acknowledgements. So the first acknowledgement that I want to give is to you, the Colorado Mycological Society. Like Alexis mentioned, I petitioned the Colorado Mycological Society for some money, and they were able to give me money to do my sequencing. So huge thank you. Also, thank you for coming out. For people who helped me do my collections, Tay Bright was a huge help in California. Andy's parents, Bill and Diane Wilson, were huge help. They let me stay with them, and they were on Pyrola watch before I got out there. Bella Briganti helped me with my Colorado collection. So did Sam Demirsky. My partner, Johnny Tormone, helped me do a lot of collecting, even on days that weren't so great. And his parents did some collecting and also let me stay at their house. So that was awesome. For field site assistants, Jonathan Schilling and Banning Starr were huge helps in Itasca and Fraser. And then Dan New at the GRBC was kind of our sequencing guru. He really helped me figure out how to sequence my samples. And for my committee, I wanted to thank Dr. Wilson in the back, but then also Dr. Green and Dr. Bronco, who have helped shape this project into what it is. So with that, does anyone have any questions? Yeah. Is it? Oh, pre-show. Thank you. Oh, yes.
How far apart are the two plants from each other down in the same habitat?
Oh yeah. So that's a great question. He's asking like how far apart the plants were when I was collecting them. The answer is it depends. For a lot of the Colorado ones and the California ones, they were literally right next to each other, like on top of each other. In Minnesota, I had some more issues where they were actually like 200 miles apart.
The question is how far are the fungi from each other?
Yeah. Yeah, so that could be why Minnesota was such an outlier, because they weren't even like close to each other. Yeah.
So I don't know much about biology at all.
Yeah.
Were you at that slide where you were showing like the family, the plant family that you selected those two species to study from?
Mm-hmm.
Are you trying to tell me that blueberries and ghost pipe are like closely related? I'm so interesting.
Yeah, the answer is like kind of, yeah. So they are in the same family, but like they're pretty far apart within that family. They're yeah.
Can you give an example of like another plant family? Like how much diversity do you see in a plant? Sorry, I don't want to give you annoying.
No, that's not annoying at all. How much diversity do we see in plant families overall? There's a lot of diversity, right? Like even like I'm thinking like of mycoheterotrophic, like I'm thinking of like the orchids, which are crazy diverse. You've got stuff like vanilla bean, and then also some of those mycoheterotrophic plants. So overall, there's typically a lot of diversity, but it's going to depend on the family too. Yeah.
So when you were barcoding, you mentioned that you only kind of identified plants that had 97% or higher matching.
Yeah.
Are you saying that there were a significant number of plants you weren't able to match to any record, or excuse me, like fungi that were on a record?
Great question. Okay, yes. So the question was like, I had some fungi that were 97% similar and ones that weren't. All of my hits were relatively high. So when I say like less than 97%, that could be 96.5%, right? The vast majority of them were still like in that like 80 to 90% range. And so when I originally did my original blasts, I actually referenced 2 different databases and then took the best match from those 2 databases. So tried to get as close as possible, but thanks. Yeah.
How do you distinguish between mycorrhizal and heterotrophic?
That's a really good question. How do you distinguish between mycorrhizal and mycoheterotrophic?
The fungi class. Do some of these fungi act as just mycorrhizal?
So do some of these fungi just act as mycorrhizal? I know that the fungi that are participating in this mycoheterotrophic relationship are typically the same fungi that are mycorrhizal, right? So it could be like the same russula is associating with this tree, but also this little pyrola. It could be like truly the same organism that's getting energy from the tree and then giving it to the pyrola. Does that make sense? Was that the question? Okay. Huh?
How do you know?
Oh, how do I know? That was the question. Okay. So much smarter researchers than me have used something called stable isotope analysis to look at the different isotopes of carbon in the plants. And they found that autotrophic plants typically are not enriched with heavier isotopes of carbon, but mycoheterotrophic plants are. And that's how you tell like what is autotrophic versus mycoheterotrophic. Yeah.
So you mentioned the mycoheterotrophic plants that they evolve, you said what, 50 times?
Yeah, 50 times.
And it may have been an adaptation to low light.
Mm-hmm.
Is there anything indicating that these adaptations may have occurred during times of mass extinctions when there were crazy conditions on the planets and less widespread need to develop?
That is such a good question, and I actually haven't looked into that. Whether mycoheterotrophy could be related back to like mass extinction events. I haven't seen any research about that at all, but I'm gonna look for it because that's really cool.
I mean, I don't know that you could tell from a fossil if using like DNA clocks
Mhm.
and tracing back when, the common ancestor of whatever mycoheterotrophic group, if they all kind of pointed to like similar time slices.
Yeah, no, that'd be really, really cool. I haven't looked into that at all. Yeah.
Question from the chat.
Mhm.
Do that word, plants have separate mycorrhizal relationships as well as being mycoheterotrophic?
So do the mycoheterotrophic plants themselves have their own? So
Mycorrhizal, like
yeah.
having more of like a symbiotic relationship.
So there is some research on whether or not mycoheterotrophy, like if that's beneficial to the fungi. So if you're a fully mycoheterotrophic plant, I wouldn't anticipate that you'd be able to support mycorrhizal fungi because they're the ones that are supporting you. But as far as like, is there any sort of like benefit to the fungi? That's been a topic of research quite recently. I think what is currently out there is suggesting that it might be that like these mycoheterotrophic plants are able to include some fungi and exclude others. Basically what that means is like provide like a kind of a barrier. So it's just like you associate with me or get the heck out, right? They're kind of like the mafia shaking down the mycorrhizal plants. That would kind of be more of does that answer the question?
I think so.
Just like what do they receive in return? Yeah.
You mentioned that at one point you thought that some mycoheterotrophic plants require association with termination. So I got to wonder if associations across the same species, but different stages of maturity.
That's a really good question. Yeah, because I sampled before, during, and after. Did I see any differences over the different times? I didn't see a significant difference over the times. It was mainly geography. I was really hoping for that. I think you might be able to find something if you looked at like truly different life stages, like if you looked at like seedling versus adult, but I was looking at just adults. Yeah. Yeah.
So German mycorrhizal fungi should have the same carbon footprint as the plant. Right?
Similar, yes. Yeah. But I think there are fewer studies.
Coming from different sugars, giving a different profile.
So for that stable isotope analysis, basically from what I understand is that photosynthesis tends to not choose heavier carbon because it's harder to like work with. And so the sugars that get sent down to the fungi are not ones that it like produced via photosynthesis, or like the sugars that it sends down tend to be heavier enriched. And like that's the carbon that gets passed along then to the mycoheterotrophic plants. Does that make sense? I may not be explaining this well. The plants send it down to the fungi. They don't use it themselves. The trees would send it to the fungi, and the fungi would then send it to the mycoheterotrophic plants. Yeah.
Going back to having it evolve 50 times, is that a convergent, or like did it evolve separately in different families, or is it, secluded to like, can we trace it back to one ancestor?
Yeah, that's a great question. Is like, did mycoheterotrophy evolve like once or many times? And the answer is many times. This pops up independently all over different plant families. Yeah.
Is it a trait that the mycoheterotrophy lifestyle, is that a trait that will say evolve in a group, and then some members of the group decide, oh no, we're going back to the old way of doing things? So do you have that, like some more derived plant that's nested within a group that's different than how they use the fungus?
That's a really good question. It's just like, do plants ever go back? I don't believe we've seen evidence of plants going back. I know that in the process of evolving mycoheterotrophy, plants don't actually evolve new genes. So these mycoheterotrophic plants didn't evolve a mycoheterotrophic gene. They lost the ability to photosynthesize. So I think there is a point of no return where once you've lost the ability to photosynthesize, they've lost the ability to produce chlorophyll. It's really hard to get that ability back. But I'm not necessarily sure, like when it comes to like partial mycoheterotrophy, if like maybe one of the chemophillas kind of like dipped their toes into mycoheterotrophy and then went, oh, not for me. I haven't seen much evidence for that.
Passive absorption of sugars.
Passive absorption of sugars. How the actual process works, I'm not entirely sure. I would assume it would be similar to like how nutrients are passed via mycorrhizal fungi. But I don't believe it wouldn't make a ton of sense if it took the plant energy to get energy. So I would have to think it's osmotic, yeah. Yeah.
Is there a way to tell like if the fungus is like directly working with the root versus like just happening to be there in the sample?
Oh yeah, that's a great question. So when I grabbed my root samples, I washed them off just to make sure there weren't any like incidental fungi just like kind of there. So that would ensure that it's just the fungi that are like in the root cells, like nestled in between the cells. Yeah. Would houseplants have them? I assume some houseplants would. I mean, over 80% of plant species do have mycorrhizal fungi. And it's not just big trees, right? Blueberries aren't all that big and they still have mycorrhizal fungi. Yeah.
Another question from the chat. Is there any explanation for why species may differ in their preference for isotopes of oxygen?
Oxygen, and carbon. So like, why would like, I don't think I fully understand the question.
I don't think I do.
Okay.
So with carbon, right, with any isotope, the reaction is slower than a heavy reaction.
Yeah. I guess the answer from someone in the back was like, anything that's heavier is probably going to be used less often than something that's lighter. Andy, or no, not Andy.
I didn't want to really interject, but the process of using isotopes is there's a phenomenon called fractionation, and when carbon gets utilized, taken out of the atmosphere, there's a different proportion of isotopes to regular non-heavy carbon. And when that gets assimilated into sugars, it then gets remetabolized by the plant for some cell function or just making cell wall material or whatever. But that metabolic process called fractionation, what we observe is that the plant preferentially uses non-heavy carbon, and then there's just residual leftover heavy carbon, sugars, and those get put away into storage, and a lot of times that's what gets metabolized or passed on to the fungus. So what we tend to see when we look at fungi in the environment, analyzing their isotopes, they have a lot more, so mycorrhizal fungi have a lot more heavy carbon isotope signatures because the sugars they get are heavier. And then we compare that to the plant tissue, the plants are lighter in carbon. And they're using the light carbon for all of their metabolic process and then passing along the heavy carbons. That's also what we see in mycoheterotrophic plants, that they tend to be higher in the heavy carbon isotopes than the autotrophic photosynthetic plants. And that's all of this called isotopic fractionation process that we believe is just a metabolic aspect of using isotopes, carbon with isotopes. So hopefully that helps.
Ed.
Sounds like, at least so far, that the mycoheterotrophy is kind of considered like it's this much. Isn't it to suspect possibly that, for example, you've got 20 species of fungi that it's associated with, but some of them it's just plain mycorrhizal with, and some of them it's completely parasitizing the fungus, and that it depends a lot on the various species of fungus that are associated?
Yeah, so we can definitely safely assume that like different fungi are providing different things. With partial mycoheterotrophy, I don't believe there have been studies that show that like the plants are able to give any of their carbon to the fungi. But it's pretty safe to assume that the plants are getting different things from their different fungal partners. So like maybe one is really good at sourcing carbon, and maybe one is really good at sourcing potassium. You know what I mean? Yeah, so would partial mycoheterotrophs have less chlorophyll? I feel like it depends where on the spectrum you're looking, right? Because for that pyrola sub-aphylla that they found in Japan, I feel like it's not that they have less chlorophyll, but their leaves are just small. I know like with mycoheterotrophic plants, like they do lose the ability to produce chlorophyll. But whether that's like happening prior to or happens after, I'm not entirely positive. Yeah.
How big can these obligate mycoheterotrophs get? What's the tallest?
That's a really good question. How tall could like the tallest obligate mycoheterotroph be? I don't know what the tallest is, but I do know that I've seen pine drops that are like this big. They just kind of shoot up. And then pyrola is like this big. So cool.